So here we go. Reactants, products, electron transport chain. 2 of the NADHs that are going to enter here come from glycolysis. 2 of them come from the prep steps. So this is 2 NADH from the prep steps. Six of them come from Krebs cycle. And then we also have 2 FADH2s that come from the Krebs cycle. We have 6 O2s. And I'm going to put in parentheses here because this is important. This is 12 oxygen atoms. Right? It's 12 oxygen atoms. 6 O2s, 12 oxygen atoms. People get confused by that when we start going through this, and so I just want to write it up there. And the other thing that we'll need is 34 ADP. And the 34 ADP come from cellular processes. Whenever you use up ATP, it turns into ADP, and so we needed the uncharged version ADP in order to charge it back up. Okay? So these are our reactants for the electron transfer chain. Like I said, we're going to save our products for later. So total we have 2 plus 2 plus 6, which is 10 NADH, and we have 2 FADH2. We know each of these is going to carry 2 high-energy electrons, right? It's picked up 2 high-energy electrons from somewhere else, and so each of them is going to be a pretty good reducing agent, meaning it's going to be able to add electrons to a complex, right? Okay, so let's talk about the electron transport chain. The electron transport chain, first of all, let's draw a relatively large mitochondria over here. So if you need a quick refresher, this is called the outer membrane. This is the inner membrane, right? The area inside the inner membrane is called the mitochondrial matrix. The area in between the inner membrane and the outer membrane is called the intermembrane space. Okay. The prep steps and the Krebs cycle happen in here. In the mitochondrial matrix. Glycolysis happens out here. And then finally, the electron transport chain happens embedded in this membrane here. So this is the electron transport chain. It happens embedded in that inner membrane. Now, how many total ATP are produced by the process of aerobic cellular respiration under ideal conditions? 36 to 38. We've talked about this already, I think, but just to put it out there, 36 to 38 is a range, and the reason for that is because of something that is called the pyruvate shuttle, right? Which means that there has to be a way for pyruvate, which is created out here in glycolysis, to get into the mitochondrial matrix to be further broken down in the prep steps in the Krebs cycle. In some organisms, that process requires 2 ATP to move the 2 pyruvates in, right? And so if it does, then you would actually produce 38, but you'd use 2, and so we'd say that 36 would be produced, right? That's where the difference comes from. All right, so let's look at the electron transport chain. First of all, it's embedded in a membrane. So we've got phospholipids, and it's phospholipid bilayer. protein complex. Phospholipid bilayer. These protein complexes, they could be drawn more accurately than this, but we are not going to worry about that. It's beyond the scope of the course. Four of them is what we need. And then after this, we're going to draw another protein. This one we have drawn before. It's got a familiar shape, hopefully, by now. This one, the star of the show, is ATP synthase. So this is ATP synthase. And we're going to say that these things, these ones that we've drawn that look like this, these are iron containing protein complexes. There are some specific ones in here that are called cytochromes. But the important part is to understand that you've got iron-containing protein complexes. And these iron-containing protein complexes, they are important for this process because iron, if you remember from chemistry, is able to change its oxidation state, right? So do you guys remember in chemistry that when you named a compound that had iron in it, you had to either say that it was iron 2 or iron 3? So like for instance, there's two types of rust. Rust is iron oxide, right? But there's iron 2 oxide and there's iron 3 oxide. There's FeO and there's Fe2O3, right? And the reason for that is that iron can take multiple different oxidation states, meaning it's going to need different amounts of electrons. And so whenever you reduce or oxidize iron, it can change its oxidation state, which turns out to be pretty important. So we can say that iron can change or changes between Fe2 plus, or plus 2 rather, and Fe plus 3, right? And these are different oxidation states. Okay? And each of these complexes contains a core of iron, right? And so what happens is, as the NADH, each NADH has how many electrons? 2 electrons. So we can have 2 electrons that get dumped into here. Okay? What happens when, because these electrons come from here, what happens when electrons go into a complex? And they what are they going to do to the charge of the complex? What's that? Add to the negative charge, right? So they're going to make it negatively charged. And when something becomes negatively charged and exposed to water, what happens? Picks up hydrogen ions, yes. So H plus ions are going to go in here like this. And here's the important bit, okay? So these H plus ions are going to go in here. Then the iron is going to change its oxidation state. And then it's going to transfer those electrons to the other side, right? And then these electrons are going to leave, right? And when the electrons leave, they leave from the other side here. So instead of these H plus ions just falling back out on this side, the electrons leave from the other side. And when the electrons leave, that gives it a positive charge, and it's able to shed H plus ions. Okay? So the book, when the book shows this, right, it shows that 2 H plus ions for each electron there. It's going to show that H plus ions are being pumped across, that was in a really inconvenient spot. H plus ions are being pumped across this membrane like this. Okay, but they're not really being pumped. There's no channel in here or anything like that. What happens is the H plus ions are going into a compound on this side, and they're being released from a compound on this side, right? So they're being moved across the membrane here. And so based on the direction that they're moved here, we can tell, and where the NADH was, we can tell that this space in here is supposed to represent the intermembrane space. And this base represents the mitochondrial matrix. Okay, and the reason why I want to show you here that it's not just pumping H plus ions, that it's actually just incorporating H plus ions into compound and releasing them from compound over here, is because when we get later on to this spot, it wouldn't have made any sense to you, and it still doesn't make any sense the way that the book says it, because it looks like this pumps H plus ions, but that's not what happens at all. Okay, so when we get to that, it'll make more sense. All right, so this goes on to here. This complex doesn't really do anything. The same bit happens here, where 2 H plus ions, whoopsies, are going to come into here, and then it's going to release 2 H plus ions here, which again acts like it's sort of pumping them across the membrane, but it's not really pumping them. And then those electrons go into here, like this. And here's the important bit, okay? So these electrons are going to leave. They leave not on their own, by their own will, right? They get forced out of here. They are taken from this last complex. First of all, we refer to these complexes as complex 1, complex 2, complex 3, and complex 4, right? Which is nice because it's 1, 2, and 3, and 4. It's not like photosystems where they're all crazy numbering. This is just complex 1, 2, 3, and 4. And they are removed from complex 4. Complex 4 is oxidized, right? It gets oxidized. It loses electrons. And it's oxidized by a really, really good oxidizing agent, right? An oxidizing agent is something that's able to oxidize something, right? So if we have to think about really good oxidizing agents, what is a really good oxidizing agent? Oxygen is a really good oxidizing agent. So we say that oxygen is something called the terminal electron acceptor. Terminal meaning final, electron acceptor meaning electron acceptor, right? So these two electrons here, they go into an oxygen, right? Now, does oxygen exist like this? No, right? So oxygen, when it's by itself, is what? O2, right? But what I was saying up here is that you have 6 O2s, and that actually corresponds to 12 oxygen atoms, right? And the way that you'll see this written in the book and pretty much everywhere else is that this thing here is written as 1/2 O2, right? Which is silly because it's really just an oxygen, right? So don't get too freaked out by seeing 1/2 O2 because 1/2 O2 is just O. Because it exists as the molecule O2, but you have to really have two of them, two of this process happening in order for it to happen one time, right? So it's listed as 1/2 of the reaction is O2, essentially, right? Okay, so it's about balancing oxidation reduction reactions. If you've had AP chemistry, you've written the 1/2 before, almost certainly, but it's confusing if you've only had chem honors. All right, so 2 electrons come into an oxygen, right? Those make it oxygen -1, oxygen -2. So now we have oxygen 2- here. What happens to oxygen 2- when it's floating around in water? Picks up 2 hydrogen ions. So 2H plus go into here. And when 2H plus go into there, what is it now? It's now water, right? So the reason why it doesn't make any sense to me for the book to show the H plus ions being pumped across here is because these H plus ions, they never end up over here, right? They just take away H plus ions from this side. But if all you're doing is trying to create a gradient, does it matter if you're just taking them away or if you're moving them across? No, because it's still doing the same thing, right? If we look at the total difference here, right, this makes this interior space taking away 2 plus, taking away 2 plus, taking away 2 plus. So now the interior space is -6, and the inner membrane space now is +4. So the difference between them is 10, right? So -6 and +4, difference between them is 10, the overall distance, right? Make sense? Like if you thought about it on a number line. That's all we're doing. We're creating a gradient here. So it doesn't matter if we're just capturing them or if we're capturing them and then releasing them, right? It's all the same. So the point is that now in the inner membrane space, we have a much greater concentration of hydrogen ions because we've gotten rid of 6 hydrogen ions from the inside and we have added 4 hydrogen ions to the outside, or to the inner membrane space. Right? Does that make sense? Have I lost you? Are you confused by this? Please ask questions if you're confused. We're good? Okay. So now we've got a bunch of H plus ions. Let's just say, for the fun of it, we're just going to put a bunch of H plus ions over here that we have added to this space.
Oh wait, I have a question.
Yes.
So where does ATP come in?
Aha, good question. We haven't gotten there yet. All right. So now we have a bunch of H plus ions. All that we have accomplished through this entire process, we went through glycolysis and prep and Krebs, and we made a bunch of high-energy electron carriers, NADH and FADH2. We used those electrons to create an H plus ion gradient, right? Now here's the magic. So the H plus ions, they flow back through this molecule here, this protein here, which is called ATP synthase, and we know that through the process of what's called oxidative phosphorylation, we are going to take ADP plus phosphate and make ATP. This process is called oxidative phosphorylation. Okay? Questions so far? Yeah, what's up?
So the amount of H plus ions that are going up through all the proteins, making the gradient, can only that same amount go back down through the ATP?
Yep.
Okay.
What's up?
Where does the phosphate come from?
The phosphate is just floating around. We can always assume a couple things in biology. In every biological organism, every cell that we're talking about, we are going to assume there is an unlimited amount, and this is not always the case, but we are always going to assume there's an unlimited amount of hydrogen ions in the cytoplasm. There are an unlimited amount of free phosphates in the cytoplasm. There are an unlimited amount of free amino acids, right, that are just floating around, and free nucleotides, right? Those are all just floating around, and they can be used at will. It's not always the case, but we're going to assume for the purpose of this course that that is a thing. Yes?
So NADH will give up 2 electrons. So it'll become NAD+.
NAD+.
And then that's why the hydrogen goes through. Does that mean, why does the hydrogen H plus ions go through on the first and third, but not the second and fourth?
Okay, so the second and fourth have other jobs, right? So the fourth is responsible for doing this oxidation reaction with oxygen.
So does that happen in the protein?
Yes, this protein here, really the real job of this protein is to be the oxidizing agent for this protein, right? So this protein oxidizes this protein, and then this protein gets oxidized by oxygen. Right?
So the second one gets oxidized by the first.
Not quite. No. So look, the second one doesn't do anything, right? This one has a job, this one has a job, this one has a job. So what is the second one even there for? The second one is there, great segue, thank you, because there's another molecule. A molecule that has electrons, but they're slightly lower energy, right? So this is the entry point for NADH's electrons, and this is the entry point for FADH's electrons. So this is essentially here to reconcile the energy levels of NADH and FADH2, right? So FADH2, you can see, doesn't produce as much H plus ion gradient because its electrons don't have as much potential energy, right? And so that means that they're going to pump less H plus ions, and therefore produce less ATP than NADH did. What's up?
Wait, so since like 2 electrons are going in, but then in the first one, like 2 went in, like the second, so that means there's 4 electrons in the second one?
No, so these are separate processes. I mean, they all happen sort of very quickly, but so you can think of it as like a pathway. So the first pathway would be from NADH, and that's boom, 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 right? Ending up here. Yes? The second pathway is this. Boom, 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 boom. Right? So it's just a matter of where the electrons came from. If the electrons came from FADH2, they would get added here. If the electrons came from NADH, they would be added here. Right? Okay, so let's talk about our products then. So this produced a bunch of ATP through oxidative phosphorylation, and that means our products are now 10 NAD plus 2 FAD 12 H2O and 34 ATP. Okay? Now, we had oxygen, and by having the oxygen, we made a ton of NAD+, right? Yes? Now, this is really important because we said that the reason why the process of glycolysis can happen without oxygen is because it has another way, fermentation, of making this molecule again. Because if you don't have oxygen, you can't go through the electron transport chain, and therefore you can never free up your NAD+. And so running out of NAD+ will cause you to only be able to do anaerobic respiration, right? So ultimately, what causes your cells to go through anaerobic respiration? Lack of oxygen, which then causes a lack of NAD+. And your cells are like, well, we got to get the NAD+ from someplace. I guess we're just going to make lactic acid to get the NAD+ back so we can keep going through the process of glycolysis to keep making ATP. Right? Now, we made 12 water. We used 6 O2. That's part of the equation, right? So far in this process, we've gone through and we've used up the. So overall, we've used up our sugar. Our sugar was C6H12O6. In the electron transport chain, we used up 6O2, right? And in the prep steps, we made 2 carbon dioxide. And in the Krebs cycle, we made 4 carbon dioxide, so that's 6 CO2. And then in the electron transport chain, we made 12 H2O. Is this the right equation? No, right? And the reason why it's not the right equation is because in the prep steps and the Krebs cycle, every time we break down this carbon-containing compound here, we have to go through what's called a hydrolysis reaction, right? And when you go through a hydrolysis reaction, you use 1 water. Right? So how many hydrolysis reactions did we go through? 6 of them to break down the C6s into 6 CO2s. And so we used 6 H2O over here. And so what we do in this equation is we say, okay, well, we're using 6 H2O, but we're producing 12. So we just cancel out 6 of those and make it 6 H2O. And that's the familiar equation that we see all the time. Okay? So we often say that the carbon dioxide is a waste product, and that's true for animals because animals can't use the carbon dioxide for anything. But in a plant, this carbon dioxide is very useful because it can be used in those exact same cells to go through photosynthesis, right? Because plants are going to go through both processes. What about the water? Is the water waste? No, we need water, right? In fact, you know that whole thing about you should drink like 8 glasses of water a day? That's mostly garbage because you get the vast majority of the water that your body requires from eating, from breaking down food. Now, is it bad to drink more water? No, you can totally drink more water. And if you have a high protein diet, you absolutely need to do that because, as we'll talk about later, when you eat protein, part of this process of metabolizing protein produces something called nitrogenous waste. And nitrogenous waste has to be exported from your body. And we know that it would produce nitrogenous waste because proteins have amino acids in it, which amino acids have nitrogen, and therefore you break those down and you have this extra nitrogen, yada, 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 right? But the point is that you make all this nitrogenous waste, and the nitrogenous waste has to be excreted from your body via urine, right? Via this stuff called urea. And when you produce urine, urine has a ton of water in it, right? And so you waste a ton of water. And the more urea that you produce by eating a lot of protein, the more you have to get out of your body, the more urine you produce, the more water you waste. And so if you have a high protein diet, you need to then drink more water, right? There are certain organisms, like reptiles, for instance, that basically never drink, right? You never see them drink water. And the reason for that is that they produce a ton of water just from eating stuff, just from going through cellular respiration. So then how do they get by this whole, wasting water by urinating? Well, guess what? Reptiles don't urinate.
No way.
Yeah. We have a product called urea that gets dissolved in water. And what they do is they make their nitrogenous waste into uric acid, which becomes a big white clump that looks like bird poop. In fact, in bird poop, the white part is uric acid because birds are descendants of reptiles and they also don't drink, right? So the white part is bird pee, right? It's uric acid. It's nitrogen-containing waste that is the same as the nitrogen-containing waste that would be contained in our urine. What's up? Very rarely. I mean, like if there's a bird feeder or something, they will. Or a bird bath, but very rarely are they imbibing water. What's up?
So all reptiles don't pee.
Correct. What's up?
So do they excrete it like through their digestive system?
They have so they have a single hole, a single opening called a cloaca, right? And the cloaca is going to excrete urine and feces, oftentimes together. So it'll have like a big white chunk and then like a little black chunk on it. And the black chunk is the leftovers from their digestive cavity, and then the white chunk is all the uric acid that they're excreting. What's up?
It's known that, like, I don't know, you can't survive like 2 days without water. If you were eating during that time, you can survive that long.
Yep. Yeah, if you were eating like just pure glucose during that time, now you got to like also consider that you are doing hydrolysis reactions and stuff. But yes, you would last a much longer time if you were eating without drinking. Well, that has a lot of water in it, so you're sort of cheating then.